thank you guys for joining for another installment of Chestnut Chat. Welcome. If you haven't joined us before, just to let you guys know, if you want to tell us something or just, chat about what we're talking about, throw that in the chat. If you have a question, please put that in the Q&A module. The Q&A is a lot easier for us to keep track of the questions. If you put it in chat, we might lose it just because things shift up and we can't keep track of it real well. Today, I am thrilled to be joined by Mark Double from WVU and Laurel Rogers from Shenandoah University. We're going to talk about Cruddy Bark and the complex world of chestnut cankers. A couple more housekeeping things. If you're not one of the four people here in the panel, Mark, me, Laurel, or Lisa, we can't see you, we can't hear you. So feel free to talk on the phone, eat lunch, do whatever you do. It's not going to interrupt us. But hopefully you guys will enjoy today's program. With that, I'm going to turn it over to Lisa to give a little bit more of an intro for today's program. And then we'll turn it over to Mark and Laurel.
Great, thank you, Sarah. And for those of you that don't know Sarah, she is the Director of Restoration for the American Chestnut Foundation, our 20-year veteran. So she knows just about everything there is to know about chestnut and is my go-to person very, very often. So thank you, Sarah. And she is definitely the owner of these Chestnut Chats, which have created a kind of a virtual community of its own, and we're thrilled about that. Thank you for everyone for joining us. I'm Lisa, I'm the president and CEO. And with that, I won't stall any longer. Mark and Laurel, thank you so much for joining us. Mark, go ahead. I don't have a roast plan for you as you did for me in Huntsville, but your day is coming. Just be prepared.
All right, thanks. Well, welcome
Sure.
everybody. Today, Laurel and I are going to talk a little bit about the complex world of cankers. And I have a disclaimer at the beginning that when Sarah Fitzsimmons called me probably around Christmas to ask me if I would be interested in doing this Chestnut Chat on cankers, I asked her what she wanted me to talk about. And so she emailed me a list of questions that she thought people would be interested in knowing the answer to. And so all I have done is taken Sarah's questions and filled in the blank with some pictures. So this talk is basically Sarah's, although I'm going to be the mouthpiece along with my pictures. So I'm going to talk a little bit about cankers, and then Laurel's going to piggyback with her talk about her microbiome research. So for those of you who are new to chestnut or new to the Chestnut Foundation, what is chestnut blight? Chestnut blight is a fungus. It's a microorganism. And when we think of microorganisms, we think of bacteria and fungi and viruses. And viruses are certainly in the forefront of today's world with SARS-2 coronavirus. But chestnut blight is a fungus. It's a canker-causing organism. And you can see on the picture on the left, which is a young, susceptible American chestnut tree, which has all those little orange bumps on them. And they're orange because the fungus, which is Cryphonectria parasitica, is pigmented orange, as you can see on the Petri dish on the right. So I want you to pay particular attention to all these little bumps on these chestnut blight cankers. And this little dead branch, this little twig, this is quite often the source of where a chestnut blight spore will land and initiate a canker. So that canker then will begin, that fungus will begin growing out from that original area where the spore landed. And quite often those are dead branches, which makes a good entry point for the fungus. So how does the fungus spread was the next question Sarah thought you would be interested in. Well, there are two different spore types. And the first we want to talk about are asexual spores. And I'm not going to delve into the complex world of compatibility, but there are 64 known compatibility types of the fungus. And when you have an asexual form of the fungus, which are produced quite often in these real sticky pig-like tails, when the weather's nice and moist, there's high humidity. These are all the same compatibility type. All 100,000 or however many spores are in these tendrils. And you can see in the picture on the right, I took this on a moist day, a cloudy day with it just finished raining, and you can see quite clearly these little pigtails that you see over here as a close-up. And this is most often associated with localized spread. So you can see how rain could wash down a canker and wash over these tendrils and carry the spores down the stem, or as insects crawl up and down the stem, chewing on these tendrils and walk up and down the tree, that they could carry with them the spores of these asexual form of the fungus. These asexual spores are called conidia. And those are in contrast to sexual spores. And remember I told you, look at those orange bumps on a canker? Well, I've taken a plug from a canker, and you can see all these orange bumps. And when you take I've lost the word, a knife, and you cut off the tops of these are called stroma. See all these black dots that are protruding from the midst of this orange stroma? Well, those correspond to these necks. So these structures that sort of look like a pear with these long black necks are in the midst of this orange stromal mix. This is all fungal material. And if you take a needle and you stick it into this piece of bark, after this is nice and moist, these things will pop right out with the needle. And I've shown over here on the left, the entire underneath section of one of those orange bumps. And you can see these structures. There's 1, 2, 3, 4, 5 that are visible. And here's that orange material at the top. And so as these necks protrude, like you can see over here, and what's inside these cream colored flasks are spores, or sexual spores. And I've got a cartoon here. They're in this sac. In this sac is called an ascus. And so quite readily then these spores, these two-celled spores, are called ascospores because they are in a cell called an ascus. And when you squash one of these structures are called perithecia. And so when you squash a perithecium on a glass slide and look through a microscope, you see all these thousands and thousands of these asci that are inside each one of these perithecia. And what happens then when it's generally cooler in temperature, these perithecia will pulsate, and these thousands of asci are shot out of these black necks and picked up and carried by the wind. So these are generally considered long distance propagules. So we have asexual conidia and sexual ascospores. This is a quick little diagram of what these orange bumps look like. You can see diagrammatically how these are buried in that orange material. This is the asexual pycnidia, which is what this is called. And these are both in the same orange area. They're both produced, the sexual and asexual spores are both produced in that same orange bump on the surface of the canker. Here's a picture of the asexual conidia to show you how much smaller they are than the two-celled sexual spores. Both of those spore types are capable of causing disease and creating cankers. So how does the fungus grow in a tree? Well, I think most of you have experimented with germinating seeds. Maybe some of you with chestnut seed. And you know they produce a radicle. Well, that radicle grows as a root, grows and then produces primary roots and then secondary roots and then root hairs. And so that's somewhat analogous to what the fungus does in the tree. So once that spore lands and the conditions are right, and that the temperature is okay and there's enough humidity, that spore germinates much like a seed. And as that spore germinates and begins to grow, produces these filaments. And those filaments then grow in the vascular cambium, where the xylem and the phloem are located. And what happens as that fungus begins to grow in that vascular cambium, it produces organic acids. And the most common acid is oxalic acid. And I've taken a young stem here and cut away the barks to show you this brown infected area, which is where the fungus has already killed the tissue, as opposed to this nice clear white area of the tree, which is yet to be infected. And as this fungus these filaments grow through the fungus or grow in the vascular cambium. They're producing this oxalic acid, which is pumped out of those filaments in advance of where the fungus is actually growing. And it's the oxalic acid that's actually killing the vascular cambium tissue. And so what happens is the fungus then not only grows lengthwise, it also grows around the stem. And as it grows around the stem, all the tissues that transport water are then killed. So everything distal to a canker is then killed. And that's very evident in this clump sprout picture here on the right, where you see all these brown leaves. So that's indicative that at the base of that branch, there is a canker that has girdled that branch, cutting off all the nutrients and water to those leaves. And that's when they turn brown. So that's what was evidenced about 120 years ago in the large trees in New York City, when the fungus was transported on infected rootstock from Japan into the port of New York. And those spores of the fungus found a species of chestnut that had no resistance, because as the chestnut blight fungus and Asian chestnuts grew together over millennia, Asian chestnuts developed resistance genes to the fungus. That doesn't mean that Asian chestnuts don't get infected, they do, but Asian chestnuts can ward off the infections and continue to grow. Unlike American chestnuts and European chestnuts, which are highly susceptible to this fungus. So this is what they saw. This is called flagging. And when they saw that in New York City, they realized there was something terribly wrong with their large chestnut trees in the Bronx Zoological Park. I'm going to flip right into hypovirulence. So we've gone from an orange colored fungus to this white pigmented fungus. So hypovirulence is a part of the 3 legs of the American Chestnut Foundation's 3-burr program, 3 Bs. The Bs are breeding, biotechnology, and biocontrol, which is short for biological control. And the UR is united for restoration. So 3-burr, breeding, biotechnology, biocontrol, united for restoration. So this white fungus that has this orange yellowish center is the same fungus as we saw earlier in the Petri plate, where the whole thing was orange from one end to the other, from one end of the plate to the other. This is the same fungus, but it is infected with a virus. And so what does the virus do in the chestnut blight fungus? The virus reduces pigmentation. And you can see that instead of a lot of orange, it's mostly white. We call this the fried egg effect. So you have a nice orange center with a white periphery. It also reduces the sporulation of the fungus, its ability to produce those sexual and asexual spores. Because when the plate was on a plate that's orange, there's spores being produced throughout the entirety of that colony on a Petri dish. That's not the case in this white form of fungus, because the spores are just produced in this little area that's pigmented. So it reduces pigmentation, it reduces the ability of the fungus to sporulate, and it reduces the ability of the fungus to grow in a tree. The picture of the tree on the right with that scaly looking bark is a European chestnut, which is also susceptible to chestnut blight. But this canker looks very, very different from the canker I showed earlier that was a killing canker. This is a very superficial fungus. It does not kill the tree, and the tree can continue to grow. So this is hypovirulence. So how do we treat trees with hypovirulence? A lot of Petri plates are grown with that white form of the fungus in the laboratory. Those Petri plates are then the mycelium and the agar are ground up in a blender. That blender is then we have like a soupy mix that is added to a sterile squirt bottle. We take steel punches and a hammer, and we try to ring around the margin of a canker. And we use that hypovirulent mixture to squirt that material into the holes that are then covered with tape to retard the drying of that material. And we're looking for this sweet spot, this picture in the middle. Here, the dark area is the infected area where the fungus is growing. This lighter area is uninfected. So that's the spot where we're aiming to put this hypervirulent mixture right into that hole. And if we're successful, you end up in a year or two with a canker that looks like this, and that the callus tissue closes over that canker. So remember, the hypervirulent form of the fungus reduces the ability of the fungus to grow in a tree, and the tree can utilize its own defense mechanisms by producing callus to wall off that infection. The question becomes, can this treatment be used to stop all cankers? And the answer to that is no. And we'll get into something that's relatively complex, but I'll try to make it as simple as possible. If you take a cube of fungus from a petri dish from the orange virulent form and place it somewhat adjacent to a cube of fungus from the white form of the fungus, and allow them to grow together over about a 10 to 12-day period. This plate on the right, the orange form grows nicely and the white form grows nicely. There's no interaction between the two. As opposed to the petri dish on the left with the orange cube and a white cube. And what happens is right here, the orange fungus begins to grow white, meaning that this orange fungus has acquired the viruses, or the virus. And so I've tried to show that here in this little diagram, in that this filament of the fungus is from the orange, and this filament is from the white form of the fungus, and these little dots represent the virus. And if you have the same compatibility type, then those two forms of the fungus, the orange and the white, will fuse, and those fusions become functional, and that virus is then passed to the orange form of the fungus, which occurs right here on this plate on the left. So that all the growth subsequent to this orange form that is now white contains the virus. So if you are a compatible pairing, meaning that the white form and the orange form are the same compatibility type, and you inoculate that into a canker, this is about a year after these holes were made and the inoculum was added into this. There's absolutely no expansion whatsoever. It has stopped canker expansion altogether. As opposed to this canker on the right, where it's also been treated, the black magic marker indicates where the canker, the margin of the canker was where we treated, when we treated it. And you can see that the canker has continued to grow well beyond where we treated. So there's no conversion, much like the same in the petri dish, whereas there's conversion of this canker, much like this incident right here. To show you on excised chestnut stems, this is the white form of the fungus. We took some chestnut stems that are cut in the winter and took a small piece out of the top stem and inserted it with a piece of the white form of the fungus. Did so exactly the same thing with the orange form of the fungus on the stem on the bottom. After about 7 weeks in a moist chamber, this white form of the fungus grows a little bit in comparison to the huge canker that's produced by this orange form of the fungus. And in addition, remember we talked about the white form of the fungus being able to reduce sporulation. These orange dots are all sporulation that are developing from the orange form of the fungus. You don't see that up here on this hypovirulent form. So hypo means less than. So the whole idea of this white form of the fungus, it produces a canker that is less virulent or grows less than this orange form of the fungus. So how do we know if hypovirulence is in a canker? Well, initially back in the 1970s, we used cork borers and took large plugs out of a canker, and we put those in. We probably took 3 or 4 large plugs from a cork bore and put those into a sandwich baggie. And we put a small piece of paper indicating the plot number, the tree number, and the canker number in that baggie with those 3 or 4 bark plugs, tied it closed with a twisty tie, and then added more bark plugs to that same sandwich bag. So we would generally have 3 cankers in one sandwich bag, each twisted off one from the other. When we got back from the laboratory, then each one of those sets of bark plugs were all sterilized individually. Well, we came across bone marrow instruments. And back before the advent of the internet, when you could buy these things online, we actually had to go to local hospitals and get used bone marrow instruments, sterilize them in a high Clorox content for several days, and then use them in the field. It's a great tool because this is a stainless steel shaft. It makes very small 2 mm diameter holes. And we had a lot of 96 well micro titer dishes in the lab. And we quickly realized that you could put individual bark plugs from a canker into one row of a micro titer dish and use a simple index card and label, this is row A, this is row B, this is row C, and you could identify them, the tree and the canker that these bark plugs came from. Once the bark plugs are in the micro titer dish, they're secured in place with tape. This card is then affixed to the plate, and you can put these plates in the freezer. Chestnut blight is an extremely easy organism to deal with, and you can freeze these things for a long time. When you're ready to process the plates, they're taken out of the freezer, the tape is removed, the plugs are allowed to thaw out, and then we cut metal screens that are exactly the same size as the plastic micro titer dish. We affix rubber bands to make these snug to the plastic plate, and they're simply immersed into a 10% Clorox solution. We make sure that the plate is submerged in that Clorox water. We soak them for 15 minutes. We remove the rubber bands and the screen, and we aseptically transfer those bark plugs onto an agar medium that contains antibiotics, because if you don't add antibiotics, or some people use acid to acidify their media, you'd end up with nothing but big blobs of bacteria. So when you add either acid or antibiotics, the chestnut blight fungus comes out very easily. So these are 4 isolates of the chestnut blight fungus from those bark plugs that came from a micro titer dish. We are finding that over years of sampling cankers that have been treated with the hypovirulence slurry, that you have a mosaic of isolates. You have those isolates that are virus containing, and those isolates of the chestnut blight fungus that are virulent or not virus containing. Is Cryphonectria parasitica, or the chestnut blight fungus, the only fungus that lives in trees? No. Since we normally take 12 isolates, 12 bark plugs from each individual canker, sometimes, quite often, we get the hypervirulent white form. We also get the orange virulent form, but we also get other fungi that are also inhabiting the bark. This is a fungus called Pestilotia. This is a fungus, this black thing called Epicoccum. We have 2 different species of penicillium that come out from the bark plugs. One of the most one of the fungi that occurs most often with our isolations is this green fungus, which is called Trichoderma. And you can see on the plate on the left, this is the chestnut blight fungus here and here, but they have been overtaken by this very, very fast growing Trichoderma fungus. Now, if I was to try to go in and re-isolate the chestnut blight fungus from this plate, I would be unable to do that because it is now encumbered by this fast-growing saprophytic Trichoderma species. Quite often in very old cankers, when you go to sample chestnut blight, you end up with nothing but Trichoderma. And so if you were to try to attempt to isolate this Cryphonectria parasitic isolate right here, you would be unable to, because this Trichoderma overtakes absolutely everything on the Petri dish. Here are a few examples of some of the common fungi we isolate out of cankers. There's the Trichoderma, Didymastilbe, Botryosphaeria. We have a lot of different species of penicillium, Dothiorella, Umbellapsis, Paraconiothyreum, Epicoccum, and Pestilotia. And Laurel may talk a little bit more about some of these. So when you take a very young canker, a very small canker, and you take 12 bark plugs out of a small canker like this, almost always are going to yield 12 isolates of virulent form of the chestnut blight fungus. They're all orange, all 12 of them. Generally speaking, there are very, very few are there other fungi that inhabit very young cankers like this. When you take a canker that's been treated for 2 or 3 years, like this picture in the middle, you get virulent orange form of the fungus. You get a few white forms of the chestnut blight fungus, and then you get some trichoderma and some Didyma stilbae. So it's more of a mosaic as cankers age. So quite often other fungi then find that dead material suitable for their growth. Now let's move on to a canker that's maybe 11 to 20 years old. Huge callous lips on a canker that's been treated 11 to 20 years ago. You still get some virulent isolates out of this canker. You still get white forms of the fungus out of this canker. But more often than not, as cankers age, you get more and more of these other fungi inhabiting this bark on these older cankers. So sometimes other fungi can overtake the chestnut blight canker in the field. We were doing a pathogenicity study to see how large cankers were from various and sundry different isolates. And we put a chestnut blight fungus in this to allow the fungus to grow to see how large it would be. And this is all Trichoderma growing over top of that canker naturally in the field. Now I want to talk a little bit about Matt Culp's work as I finish up here. Matt Culp was a PhD student at Michigan State University, and his PhD dissertation was strictly around what role other microorganisms play with the growth of the chestnut blight fungus. And Matt used a lot of different fungi, but what he examined was when you put the chestnut blight fungus, and in this case, these are all isolates of Trichoderma. When you put a plug of the chestnut blight fungus and a plug of the Trichoderma on the same plate, like here and clear over here, how do those other fungi interact with the chestnut blight fungus on a petri dish? Well, you can see quite clearly that the Trichoderma is overtaking the Cryphanetra parasitica. This is a virulent isolate, not a hypovirulent isolate. So this should grow and be able to kill a tree. But this Trichoderma is growing quite easily over the chestnut blight fungus, and after another day or two, you won't be able to extract the chestnut blight fungus from this petri dish. So Matt came up with this conclusion. What role do other microorganisms play? And Matt said they portray hypervirulence treated cankers as a mosaic. And so you can certainly see there's a mosaic of different fungi, Cryphanetra and other fungi in a canker. His data suggests that canker communities are not stable, and volatility of the fungal community indicates that cankers can change quickly from less severe to more severe cankers through the loss of hypervirulence. So less severe and more severe is less severe is that you have more white forms of the fungus, and more severe means that there is more orange forms of the fungus. So non-Cryphanetra, these green fungi, may facilitate canker expansion through greater inhibition of the white forms of the fungus relative to the orange form of the fungus, which may permit the virulent or orange C. parasitic to escape hypovirus infection and resume rapid canker expansion. So for many, many years, we knew that these other fungi were isolated from cankers, but we did not know what role they played. And so I think Matt Culp has definitively shown that these other fungi do have an impact. And I think Ursula Heinecker, if she's on here from Switzerland, can state that they found the same thing in Switzerland, is that sometimes the cankers that are inhabited mostly by white C. parasitic can also then change to orange form of the fungus, making them more a killing form of the fungus, and the canker can continue to expand. Now, the title of this whole thing was Cruddy Bark. Well, I've not worked a lot with Cruddy Bark. Cruddy Bark, for those of you who don't know, is evidenced by this picture on the right. This is a canker in Maryland. And these Cruddy Bark cankers have these huge expansions of callus tissue. Now, this doesn't look anything like a virulent canker. It doesn't look anything like the hypovirulent cankers that we've shown from Europe. but just these massive amounts of callus tissue. So we've sampled some of these cankers in Maryland, probably in 2015 or so. And we had a high school student from Poolesville High School in Maryland, Marlene Hagblade, who also sampled cankers about a year later. And our data were similar. Of all the plugs we took out of these cruddy bark cankers, about 10% were the orange form of Cryphonectria, 10% were the white form of the Cryphonectria, but 80% were other fungi. So Matt Culp from Michigan State came down and did a pretty extensive sampling of these cruddy bark cankers. And he found almost all of his isolations were not Cryphonectria, but other fungi. So what was the most commonly isolated other fungus? Trichoderma, that green fungus. So Trichoderma is an extremely fast-growing fungus, and it can be used a lot in soil drenches to try to control other fungi. So they bought a commercial blend of a Trichoderma root drench to see if they could duplicate this in the field in Maryland. They could not. So there must be something endemic to the soil microbiome of these trees in Maryland that is the cause and effect of these cruddy bark cankers. This is my last slide. This tree is planted in West Virginia, not Maryland. This is a tree arose from nuts collected by Dr. Gary Griffin from Virginia Tech. And his specialty is large surviving American chestnuts in the Appalachian region. And this tree resulted from a nut from a large surviving American chestnut that Gary found. This tree's never been treated with hypovirulence, but you can see the same sort of massive callus tissue on this tree from a large surviving American that is somewhat similar to those cruddy bark trees from Maryland. So once again, this has not been treated with hypovirulence, but it exhibits the same type of effect. Now, whether there's any more resistance in those Maryland trees, we don't know, but there's certainly something going on with the microbiome of those cruddy bark cankers in Maryland. And with that, I will stop my share and let Laurel take over.
Thanks, Mark. Before Laurel starts, though, that was excellent. Thank you. And I also love your photographs. Those are phenomenal, especially the all 9 of those together.
They were your questions, dear. I filled in with the pictures.
Well, they're fantastic. How long does it take you to learn how to pronounce all of those fungal names? I think
not very long.
Okay. Well, very well done. I'm going to ask you a couple questions before Laurel starts, because there are some really cool questions in here that I want to get to. Kent Wilcox asks, can pure free virus, free of the fungus, be isolated? And if so, will that virus infect the blight fungus, or is infection of the virus dependent on compatibility type and solely via cell-to-cell contact?
I'll stop you right there. The answer is no. You can't. The virus does not live outside the fungus, so you cannot free the virus from the fungus. You can't free the fungus from the virus from the fungus. Sorry, I got that all backwards. But no, the virus does not live outside its host.
An obligate.
Right.
Yeah. Emma Ehrlich says she's a mammal person. What would happen if you approach this like a tumor and skin grafts? For example, cut out blight affected bark, sanitize the site somehow, cut smaller strips of healthy bark, and then patch them in the hole. Would the canker just come back because sterilization isn't possible?
Sterilization like that is difficult because remember, you're working in the forest. And so you have all kinds of things blowing around. There's all types of critters crawling up and down trees. So I think that's a good idea, but I don't know that you could do that sterilely enough to make that happen.
And I have one more question. There seems to be a lot of questions about these other things and how much they can impact being able to be cultured, how much they can be reapplied to chestnut cankers to stop them. And I mean, that's the whole point, right? Can we culture this, figure out what might be antagonistic to chestnut blight? Do you have any sense of what, like, I think Trichoderma has been implicated, a bacillus has been implicated. Can you speak to what we know about that?
Okay, so Terry Tatter back at the University of Massachusetts, and this is back in the 1980s, did work with Bacillus subtilis, which is a bacterium, and Trichoderma. And he published a few papers on that. And so one of the things when I started off earlier talking about microorganisms and talking about bacteria, fungi, and viruses, bacteria are something that we have not delved into. So what role bacteria have, I mean, Terry did some work with this Bacillus subtilis isolate and working with Trichoderma to sort of overwhelm chestnut blight cankers with Trichoderma. And that's probably possible if you get cankers that are small enough. And I think, so one of the things, let's talk about this real quickly, is that the chestnut blight fungus does not affect the root system. And so if it affected the root system, we'd be dealing with an extinct tree species. But since it does not affect the root system, chestnut has this great ability to sprout from the root collar and produce lots and lots of root sprouts. And so back in the day when they were doing mud packing, they were looking at a variety of microorganisms that were in that mud that they were applying to a mud pack. And it was this cacophony of other bacteria and fungi, and also the reduction of oxygen, because fungi are aerobic organisms. And so when you put this mud pack on a tree and you cover it over with this plastic bag, you're reducing the oxygen potential for that fungus. And so in concert, you're looking at microorganisms producing secondary metabolites that would act on the chestnut blight fungus, but also, as importantly, the reduction of oxygen. So you could probably take Trichoderma solutions and pack those onto a tree and reduce that canker. That, however, is not going to have any impact on the rest of the stem. So you may stop that one canker in its tracks, but that leaves the rest of the tree just as susceptible.
All right. I think that's a good time to switch over to Laurel. And I think there's a few questions in here about using sequencing to find out what else is in there. And that's exactly what Laurel's going to talk about. So I think that's the perfect segue. Laurel, if you would take it away. And then again, if you have more questions, put it in the chat. Kendra is typing away with answers she can do. I'm answering whatever I can, but we'll hold the questions until after Laurel. There's at least 15 more in here that we'll attack after you're done.
Okay. Well, thank you very much for inviting me. And I appreciate the opportunity to kind of talk about what project I'm working on and have been working on off and on for years. And, you know, I don't have a complete project, but I have a complete hypothesis and ideas, and I'm excited to share them with you where we're at right now and where we think our research might fit into the big picture. So I'm really interested in this microbiome concept and how it might apply to plants. And as you know, Mark was talking about with what else is happening within cankers, I'm also interested in what's happening before the canker ever gets there. So I'm only going to spend a brief time on the introduction because Mark did a great job of kind of walking you through the American chestnut infection biology. But you know, we know that you're all here because you're familiar, at least with American chestnut, understanding that it used to be a really large tree that had was really important for our ecosystems and our economy and lived up and down the East Coast. And then we ended up with the infection of Cryphonectra parasitica. And you've seen some pictures now of this fungus growing in a culture and what those bark cankers look like when this fungus gets into the tree and starts to damage the tissue. So my little life cycle drawing is not nearly as cool as the pictures that you've seen. But when a spore lands on that tree and the fungus grows down into the tree and it's surviving underneath the bark where that living tissue is, and it's producing the toxins that ultimately kill the tissue. Well, there's other things growing there too. And it's not, the tree itself is not a sterile environment, just like our own bodies are not a sterile environment. So I'm really interested in what's happening within these trees before a fungus comes, and could that provide us any clues to resistance within trees? Okay, hold on, there we go, there's our pictures. So today we do have some large surviving trees, and we do have many trees that survive with resprouts. This tree here has particularly large resprouts, but we know that many trees are not surviving. And some of the differences between trees are genetic differences. Some of these differences between trees that survive and don't survive have to do with maybe they happen to be growing in a patch of the forest with hypovirulence, or maybe there's something else going on. And I'm trying to understand the entire ecology of the American chestnut tree in order to see if there's anything else that we're missing in our explorations. So, Mark talked about the three areas where most research is focused: the hybrid trees, our genetic engineering, and hypovirulence. And I don't think endophytes would overcome any of these. I think there's lots and lots of good research into these areas that are very promising. But what I'm wondering is, our endophytes, which are beneficial fungi and bacteria, could they add a little extra level of assistance in our attempts to reestablish American chestnut trees? So plants have a microbiome, just like humans have a microbiome. So we have bacteria and fungi growing within our bodies, mostly within our intestinal tract, that are outcompeting harmful bacteria and pathogens within our own bodies. They're helping us with digestion. If we eliminate our own microbiomes, we start to have lots of, we have many health risks. The more and more we study plant microbiomes, the more and more we're coming to understand that plants exist in the same situation. We're not just within soil, where a lot of research has been done with fungi and bacteria that assist in plants with nutrient absorption. There's also a lot of microorganisms growing within the plant itself. And many times they're between the cells, they're within a tree, they're underneath the bark, they can even be found within the leaves. And they appear to have benefits to the plant itself and maintaining its health. Some studies that have happened in the last couple of decades have indicated that there could be as many as 50 to 100 to multiple hundreds of different microorganisms growing within completely healthy trees. Some of these microorganisms have been identified as kind of opportunist fungi that kind of hang out, and then once the tree dies, take that opportunity at that point to start feeding on the trees. Some of them are pathogenic but being kept at bay, but many of them, through various studies, have been indicated to actually be providing benefits to the organism that they're living in. A lot of work has been done within agriculture that have found that treating with beneficial fungi, which are the endophytes, can actually reduce damage to trees and crops. The trees that have been studied, that I've seen studies on, have been on like cocoa trees. There's been a couple incidences where they've been able to control pathogenic fungi on cocoa trees by using a beneficial endophyte. There's some of these endophytes produce toxins that are not harmful to the plant, but harmful to other pathogens. And sometimes, like Mark was talking about within these cruddy bark cankers, it's a simple and out competition situation where if you have a fungus that doesn't kill the tree, but happily lives within this tree, it can just be taking up resources and out competing Cryphonectria. And I have my own plates here of examples, just like what Mark showed you, of that when you have C. parasitic on a plate. In this particular case, this was contamination. I was not trying to culture multiple fungi on the plate. But once the contaminant got in there, the contaminant very often outcompetes Cryphonectrica. So Cryphonectria doesn't appear to always be able to compete very easily against other fungi, which led me to my ultimate hypothesis that maybe there are differences between trees with and without resistance that lead to differences within their microbiome. There we go. So that leads to my question of, when you compare an American to a Chinese chestnut tree, which the American chestnut tree has very, very limited resistance to C. parasitica, but the Chinese chestnut tree has significant resistance. Are there differences within the microbiome? And if you're even to look at, say, hybrid trees with different levels of resistance, are there differences within the microbiome? And are those differences contributing at all to the differences that we see within resistance? And if they are, can we do anything with this knowledge to enhance our ability to help American chestnuts survive? So the study I've been doing is to compare the fungal microbiomes. So while endophytes can be, and the entire microbiome is both bacterial and fungi, I'm specifically looking at fungi. That's going to be our starting point. And comparing those fungal microbiomes between Chinese American hybrid chestnut trees that are growing in the same location. So in early studies, when I was first exploring this idea, I was more interested in, how many fungi can we isolate, and getting our techniques down well, and not so as much focus on the big picture of we need to have them all in the same location. That's what we're working on now, is if these trees are all in the same place, therefore they've been exposed to similar spores within the air around them, are the trees then providing different habitats, essentially, and microhabitats for the fungi. So this big study started in 2018, and we are still plugging at it because COVID shut us down for about a year. We weren't allowed on campus for a while, and then I teach an undergraduate institution, so getting undergraduates geared back up for research has been slow. So we started in 2018. We should be finishing this summer when we're full force and a functioning lab again, where we tagged along to some American Chestnut Foundation sites where they had hybrid trees out that they needed to assess these hybrid trees for levels of resistance to figure out which ones they're going to be maintaining for breeding. And within all these plots, they always have control trees. So there's some pure Chinese trees on site, and there's pure American trees on site. So we were able to get samples from the American, the Chinese, and the hybrid all in one location. And then we did 2 different sites because we were curious to see how much difference we might see between sites as to what could be living within these trees. So the idea is, so when they go in to inoculate trees in order to test the trees for resistance, they remove a bark plug and put into it an auger plug with C. parasitica. I wanted that bark plug because I'm interested in the healthy tissue. I want to know what is there before infection ever happens. And then when we go back later and do some comparisons of how much resistance these trees had and what was their microbiome makeup. So we soaked the surface of the healthy bark with alcohol, ethanol, in order to sterilize it, and then use sterile tubes to remove the plugs and put them into tubes as quickly as we could. And then inoculate the tree with C. parasitica so that the American Chestnut Foundation can continue with their study. The rest of our work is done in the lab. We are predominantly a lab biologist. Once we get back in the lab, these plugs, and in these particular images are wedges from a different study, but it's the same concept. Just imagine a round bark plug. We place these plugs into a completely nutrient-free auger so that we can watch individual hyphae growing out from these bark plugs and isolate those hyphae and then put them onto the nutrient auger. And that allows us to do a pretty good job of getting isolated fungi. Every now and then we might get 2 fungi growing on the same plate, but we've become pretty good at catching it early enough that we can remove individual hyphae and get pure cultures. So once we have these pure cultures, we are using DNA. We would isolate DNA from each culture and do sequencing. I saw on the side screen someone asking about PCR primers. We're using ITS primers. It's one of the common ones used for sequencing and identifying fungi. It sits down within conserved regions of the genome and sequences into a somewhat less conserved region so that you can identify individual species of fungi. We also periodically will go back and confirm our results morphologically. So if we have a confirmed hit for one species of fungus, we'll go into the literature and find pictures that other people have from culturing that same fungus and make sure our pictures match. And then we also sometimes will key out based upon spores. Not all fungi form spores within culture, but some of them do, and then we can kind of key them out and make sure that morphologically what we're seeing matches up with our sequencing. And we've been pleasantly surprised that so far our sequencing has been pretty accurate and matched up with what we would have been able to identify morphologically. We get a wide range of pretty colors, pretty morphologies of these fungi, slow growing, fast growing. Typically the slow growing ones are a little bit harder to get DNA from, but we've worked on different methods in order to get DNA out of these. So what do we know so far? I didn't want to spend a huge amount of time going through the data we've collected because it's an incomplete set. So I don't want to make too many conclusions, but we have found overlap between species. So American, Chinese, hybrid trees, all have, there are some fungi species that are growing on all of them. And then we have also found differences in which there are some fungi species we're finding on, say, hybrids, and we haven't found it yet in the Chinese and American. We're about halfway through our samples, so we haven't finished yet, but there does appear to be some clear differences. And some of the fungi species that we have found and identified are popping up as being endophytes in other plant species. So it doesn't mean it's endophyte for the chestnut. At least one on this list pops up in literature as both a parasite in some plants and an endophyte in others. So a fungus can be beneficial for one plant, harmful for another. So, the next steps become complicated. But the idea is once we finish this study and we have this good map of what fungi within a single field, what are the differences in the microbiomes? And then we can look at that and start to see if there's any potential places to look at for endophytic fungi, and something that might help these provide resistance to whether it's outcompeting Cryphonectria, producing some toxin that might be harmful for Cryphonectria, that we could then use in conjunction with the other methods of breeding, genetic engineering, hypovirulence, combine these methods together in order to boost survival rates of the American chestnut. There's quite a bit of evidence within agriculture of being able to use endophytes to outcompete pathogens. And as Mark has said, we've seen evidence within cankers of some fungi that can outcompete Cryphonectria. So there's also been some study, there's one study that my students found in pine trees where they were actually inoculating the seeds with an endophyte before they were being planted, and found that pine trees had higher survival rates with the endophytes that they were inoculating. I did have one student who was really interested in when are fungi getting into these trees? Is it all after they sprout? Is it before they sprout? And he did a really intense surface sterilizing of chestnut seeds and then cracked them open, and he was able to isolate fungi within the seeds themselves. So fungi are growing hand in hand with plants from the very, very beginning. And while many of them do enter into a tree after they're growing, so many of them are going to be there from the very beginning. So there's lots of clues in previous research as to different ways in which we might be able to go once we have a list of fungi to start testing on our American chestnut trees and hybrid trees. And that's what I have at this moment in time, giving another year, and we will hopefully have a lot more clues to talk about.
That's fantastic. Thanks so much, Laurel. There are so many questions here, so I hope we can get through them all. Let's go with the last one that just came through from Gail. Do you know if any of these fungi are infected with viruses, Laurel, that you're pulling out?
I have not looked at that. We do. Yeah, I haven't looked specifically to know if any of them have viruses with them as well. I avoid the bacteria. I have not looked for viruses.
There's a question in here, and I want to add my own. Thomas asks, has anyone experimented with parasitic mushrooms like lobster mushrooms, which only grow on other mushrooms, to see if they might overtake the chestnut blight fungus?
I have not.
Me either.
I'll add in another, which I get asked this question maybe once a year, if not more often, about chaga mushroom. Apparently there was a book that came out a few years ago that recommended using what's called a chaga poultice, taking chaga mushroom, making a poultice of it, putting it on some sort of vehicle vector that you just been, much like a mud pack, slather on the canker, and apparently that's supposed to arrest the chestnut blight.
Well, let's talk a little bit about that. I mean, a lot of people say, well, can you take the Trichoderma spores and spray on the tree, or some other kind of chemical to spray on the tree? We have to realize, though, that the chestnut blight fungus is growing underneath the bark in the vascular cambium. So anything you do on the surface is going to have very, very little impact as to what happens, many millimeters down below the surface of the bark. So a lot of those applications that people think might be good will have absolutely no impact on the growth of the fungus, because that fungus is growing so far beneath the bark that now when you talk about chaga and introducing a slurry, well, I think it may be the chaga mushroom, but it's also the reduction in oxygen that may have the impact. But just spraying something on the surface it may kill the fungus on the surface, but it's going to do absolutely nothing for what's growing in the vascular cambium.
Yeah, there was a couple questions about that too, about applying trichoderma and things like that, just flooding an area. Eric Carlson, who's at ESF, I know he's looked at mud packs quite a bit. He mentioned in the chat that they did some experiments looking at mud packs and then looking at just, covering the wound. And he said that you had a greater impact using the mud pack with the covering than just with the covering. He didn't mention what the effect of just a covering. It looks like it does have an effect. So there does seem to be an interaction there.
There are a lot of microorganisms in the soil, actinomycetes, for example, where we get a number of our antibiotics. They're producing a lot of what's called secondary metabolites. And these secondary metabolites, like penicillium, or what we're using to control disease, are produced as secondary metabolites. And so when you add all of those microorganisms in a mud pack or some kind of a poultice that you apply to the tree, there's all kinds of things that are interacting, but again, it's got to be the reduction of oxygen, because this fungus is growing so far beneath the surface of the bark to have any kind of impact on the growth.
Yeah, I think one thing that I think is also treating individual cankers can be very useful if you have a tree or even if you have an orchard. But when it comes to trying to do something that's going to be forest wide, something that treats an individual canker might not end up being practical, which is why, part of what we're looking at is there anything we can do like during a planting to establish some level of assistance to these trees to get a pre-established microbiome that might be an assistance. There's definitely, with all the research out there, there are things you can do to an individual tree, but then we also need to know what can we do to a forest. Or to help maintain that forest, because we can't be going out to acres and acres and acres of trees.
Well,
there was a question about could you plant seedlings in a pot that had hypovirulent spores in it, and I don't think that's functional either, because you're relying then on the root system to uptake that fungus, and I don't think that's going to happen.
And I had a question here. And I get this question all the time. What about fungicides? Can I just treat with a fungicide? And to date, I don't think anyone has definitive proof of any fungicides working. There are some that are putative in terms of their control of control of chestnut blight. Even if they do, though, they're not labeled for use on chestnut blight. Is that correct, Mark?
Well, I think so. And I don't really know anybody who's really done that. You'd have to use some kind of systemic fungicide where you inject it through the root system or something. It's taken up through the entire tree. And I don't know of anybody who's working on that.
I know Tom Scieli is going to work with Steve Jeffers. They're going to try and implement some experiments this summer. So looking at some of the more promising fungicides, and hopefully we might have an answer at the end of this season or next season. I know a lot of people were using things like agrifos, and then they were using Bark to get that through the bark, and that seemed to have limited control, maybe 2-3 years, and then that would stop having any control of chestnut blight. So there are things, but nothing that works long term, at least not yet.
Wasn't agrifos mostly used for Phytophthora?
Yes, but it also worked with blight for a limited amount of time.
Okay.
Let's see. Has anyone? Oh, okay. Thomas also asked, has anyone ever experimented with grafting sections of bark from blight-resistant trees onto sections which do not have blight? I'll say yes, and it confers no help, because that union or whatever you graft onto, whatever's susceptible is still susceptible. The resistance doesn't travel. It itself is not systemic. Steve asks, we've heard your talk that a dead branch is a good location for entrance of chestnut blight into the xylem or phloem. What is your opinion about early removal of a dead branch followed by application of something like lac balsam to seal the wound?
I don't know anything about lac balsam to seal the wounds. A lot of times tree people were said, oh, well, here's a black tar you can use to paint over your wound. Well, actually, the black tar probably did more damage to the tissue of the tree than just leaving it alone. So I don't know anything about that application. If it's not harmful to the tree tissue and it's relatively superficial, then that might work. Because dead branches are good sources of infection courts.
Yeah, and so are pruning wounds. I know I just got a report from Stephen Hoy, who works with me here at Penn State. He went down to an orchard where the manager there had pruned a ton. Now he was pruning right in the height of the season, and pretty much every pruning wound right there had become infected with chestnut blight.
Yeah, summer is not the good time to prune chestnut trees in the midst of blight.
Yeah. Mike Alcott asks, any idea of the population of the blight fungus in forests today where there are limited or missing populations of chestnut, but probably some scarlet oak, post oak, sumac, or any other trees that might be harboring the fungus itself?
Well, in order to do that, you'd have to be a forester who's out in the forest every day to look for that. And so I don't really have an answer to that question.
Laurel, any idea?
I mean, I know if you plant American chestnut where there's no other chestnuts around, eventually it's going to get blight. So it's circulating, but I don't know at what frequency.
Yeah, it gets there eventually. I mean, even, isolated stands in Wisconsin, West Salem, of course, got infected eventually. That was probably people. But other trees that are seemingly isolated eventually get infected, whether that's local infection or.
I think you can also take into consideration how many, Chinese chestnuts are used ornamentally a lot as well. So they're going to be harboring it. So I think there's very few places that are going to be really free of blight because of trees we're using in orchards, trees we're using in ornamentals, and then there's like the post oak can still have it. A couple people asked in here about sampling other trees, and for the purpose of my study, we're staying pretty focused. At the very, very beginning, we were just trying to test methods. I did do a post oak at that point in time. Just so, I would love to do more with other trees, but in this case, I would want to see if, compare trees within a similar radius, because once you get a few miles away, you might have a completely different set of, fungi within the microbiome. So a direct comparison would be harder to do.
Well, and people need to realize too that chestnut blight has an advantage in that it's not strictly a parasitic fungus.
Mm-hmm.
It can live saprophytically. So you can take the chestnut blight fungus and live on the duff. It can live in burrs. So it can live saprophytically, so it's around a lot.
Yeah.
So it's pretty ubiquitous. Now let me make a statement here. So in West Virginia, we just found a new state champion American chestnut. It's almost 20 inches in diameter, about 80 feet tall. And it's in a very remote area of the state of West Virginia, but it was surrounded by a massive number of hemlock trees. And when hemlock woolly adelgid came in and began infecting the hemlocks and killing the hemlocks, only then did the landowner even realize there was a chestnut tree there. So now all the hemlocks have been cut down, but it's my feeling that that hemlock actually acted as a barrier for any spores might have been blowing around. And so my fear is now that the hemlocks have all been cut, it leaves that lone chestnut tree open for infection.
Mark, that's my experience, is that often trees that are isolated, now I think there's other factors as well, but I see trees that are isolated, and especially if they're kind of shaded out a little bit, once they're released, you have this combination of them growing a lot faster and having more openings for the blight to get into, but also they are then more exposed. And I find that those trees that get released for whatever reason, be it a blow over or a salvage cut like you're talking about, they get infected much more quickly than I think they would have otherwise had they been left alone.
So I'm not sure how long this is going to be the state champion.
Let's hope a long time.
Let's hope a long time. That's right.
Let's see. Bernie and Frank ask, the blight can grow on oak. So you guys addressed that, but does hypovirulence also occur in the blight that occurs on oak?
I don't know the answer to that. I'll be quite honest. I don't know the answer to that. I've never sampled that many oaks, so I don't know.
Yeah, I don't know. Eric asks, Eric Carlson, hi, Eric. Are there any efforts to engineer super donor hypovirulent strains that can spread via spores instead of mycelia? Are multiple genotypes or strains of C. parasitica being attempted to engineer into super donors, or are they all clonal?
Okay, let's talk about the super donor fungus. The super donor we currently have, it was developed by Don Nuss at the University of Maryland, produces very few spores. It doesn't even have that orange center like these the fried egg effect on the slide I showed earlier. It grows much less and it produces very few spores. The other thing is it's also regulated, and so we're not sure that it would ever be deregulated for me to send from West Virginia to Sarah at Penn State. So at this point in time, the super donor fungus has not been deregulated.
And I want to mention, so Bill McDonald has, I thank you very much for pointing this out. Bill says, someone should clarify that chestnut blight is the name of the disease, but Cryphonectria parasitica and the chestnut blight fungus, that's the name of the pathogen, only one component of the disease. The terms are not interchangeable. And he says that that's today's Plant Pathology 101 lesson. So chestnut blight is the disease, chestnut blight fungus or Cryphonectria parasitica is the organism. And I'll put another element to the plant pathology lesson. And I think, Laurel, this is applicable to your research, which is disease has three components. It has the host, in this case, chestnut. It has the pathogen, which in this case is chestnut blight fungus. And then it has the environment. And so there are lots of environmental factors that affect how much the pathogen can infect the host, how much the host can then resist the disease. We see many, many environmental factors that aren't biotic, many abiotic factors. The type of site has huge implications on how well the chestnut, how healthy the chestnut is, how much it can resist the disease. So I think, yeah, I just want to throw that out again. This is a very complicated ecology that we're working with. All right, let's see. So we got that. Has anyone experimented with growing fungus-resistant herbs like oregano or creeping thyme around American chestnut to see if that might help with resistance?
Not in my experience.
Because of the size of the tree, I would be hesitant to know if it would work, because you could get an infection 50 feet up from where your herbs are growing.
All right, so Mark Meal has a question, and I've seen this too. I think I have an answer, but I'm curious to see what your all's take is. I know of seemingly immune wild type American chestnut that has rotting wood pile at its base. All around the chestnut tree are shrubs and bushes, sprout clumps that are loaded with chestnut blight fungus, but the immune tree is a single trunker unaffected by the disease. My observation is that the rotting wood pile at the base has had beneficial effect, completely displacing the blight fungus, leaving it an intact, lovely tree without infection. Is this seen by others?
My guess is if you were to take a plug of bark out of that tree and put in a plug of virulent form of the fungus, the tree would be susceptible.
That's my guess. I have seen that, Mark. I've seen, tall individuals uninfected. This happened particularly in South Central Pennsylvania. There are tens of thousands of American chestnuts in that area, much like you're seeing in Massachusetts. And we had this gorgeous 16, 17-inch tree, completely uninfected, beautiful specimen. Must be something special about this. But no, it got infected, and once it got infected, it was dead the next year. And so I always say luck. I mean, I don't know how else to.
Well, if you look back historically at a lot of those trees that Fred Hubbard talked about for years and years that were used in the breeding program, that were uninfected for decades, and then all of a sudden they're dead. So I think it's just luck. I guess I don't think the wood pile down below has any effect on whether the tree's infected or not.
I don't think it does either, unfortunately. Tom asks, is there an upper temperature that will kill chestnut blight fungus?
Sure, I don't know what that temperature is. I'm guessing it's probably 40 C.
Yeah, I mean, there's definitely going to be an upper temperature.
Stacy Clark.
Maybe if we lived in parts of Australia where it got hot like that, that might have an effect. But in our climate here, that's not going to be an issue.
Well, I know blight is down there in their commercial orchards. They've been trying to eradicate it, but it to no avail. Stacy Clark asks, for the compatible hypovirulent cankers, how long do they last, or do they break down over time?
That's a good question, Stacy. It depends. There are some cankers, for whatever reason, that seem to be controlled and contained, and others that break over. So whatever that reason is for the breakover, we don't know yet. So it's sort of a yes and no answer to that question, Stacy.
All right, coordinating lunch. Let's see. Hypervirulence has limited, Carl Absher, hi Carl. Hypervirulence has limited ability to spread within a tree and no ability to spread between trees. So where does it come from, and how does it survive in nature?
A lot of that has to do, Carl, with compatibility types. So if you go to certain areas of Europe, and I'm not going to say all areas, but there are certain areas of Europe where chestnut blight is no longer considered a problem. And that's because when you go into some orchards or some chestnut growing areas in Europe, they would have a predominant vegetative compatibility type of the fungus. And that'd be it. So maybe if you got, let's say you've got 40 cankers in a chestnut region in Europe, and each one of those 40 cankers is sampled, maybe you find that 38 of them are the same compatibility type, and maybe 1 or 2 are different compatibility types. In contrast to the Eastern United States, where you take those same 40 cankers, you may have 25 different compatibility types among those 40 cankers. So in terms of establishment, it's much easier to establish hypervirulence in areas where there are fewer compatibility types. And so I think that's one reason that when you say, how does it get around? Well, a lot of people have done studies. I mean, Gary Griffith at Virginia Tech has done studies with mites and things like that. And Fred Hubbard's done things like that. And so there are a lot of insects that inhabit cankers, and they graze on the stroma and they crawl up and down. And so it's quite, Gary found that you can carry, or you can detect hypervirulence on mites And so I'm assuming if you used all kinds of centipedes and things, that you'd find that there are a lot of methods of the fungus that's inhabited by the virus to move from one point to another. But if it moves from a canker that's compatibility type 1, and the insect crawls over a canker that has compatibility type 2, and it's carrying a virus in the fungus it's carrying on its feet, it's not going to become established. You do the same thing in Europe, and you go from a 1 to a 1, and you pass that virus along. So there's a lot to do with compatibility and the genetic diversity of the fungus in the eastern United States compared to Europe.
So as soon as we answer a few questions, we get a couple more pop up. So I hope we can get to everybody's questions here in the next 5 or 10 minutes. If we don't, we'll try our best to get answers to you eventually. Tim Mack asked.
Yeah, I'm trying to type in. I'm typing answers to some of them.
Thank you, Laurel. Tim Mack asks, hi Tim, is anyone working on bacterial or fungal interactions on the agar? Considering that we get most of our antibiotics from fungi, this suggests that these two different clades may regulate each other effectively.
That was actually the one I was typing right now.
Okay, answer that, Laurel.
Well, what I was going to start by saying is that it's definitely some good studies to look at to do these comparisons, and doing competition assays in the lab is on our list of things to try. But I do know that there has been some studies in the past where competition assays in the lab don't always hold up in the tree. So we'll have to make sure that the assays we come up with are actually compatible, that what we're doing in the lab can actually translate to once we're in a tree. Otherwise, we might have to go straight to trying to do seedling studies.
Well, I mean, it's easy to talk about bacteria and fungi, but those are two very different sets of tools when you're working with bacteria and fungi. And in Bill McDonald's lab, we didn't really work with bacteria, but the point is that a lot of bacteria do produce secondary metabolites that might be useful, but we weren't geared up to work with bacteria.
All right, John Paul Martin has several questions. I'll see if I can get to them. Observation, you had white, orange, and green. Is Mother Nature providing the solution to the infection by introducing different players in the infection of the chestnut tree? And therefore, if we introduce different fungi and viruses, we could inevitably find a cure. I think that is more or less along the lines of what we've discussed already. Brendan asks, are the sexual and asexual parts of the life cycle produced at different times of the year?
No.
Easy enough. John Paul Martin asks again, I live in a red clay area. Clay has a long history of uses for medicine. Has anyone tried coating the fungus with a thick clay cap? And I have several questions in here about various things that you can apply to the fungus. And I think the answer is the same, but I'll leave it to you, Mark, to.
Well, I mean, when you're going to coat a canker with clay, it's essentially doing the same thing as the mud pack. Now, whether clay has the same number of microorganisms that, say, the soil in Michigan has, I don't know the answer to that. But once again, I think that the application of any kind of material that would reduce the oxygen might be beneficial to expansion of the canker. But then again, you're not going to affect any of the rest of the tree with that application of mud. So like Laurel says, if you've got a tree that's 30 feet tall and you've got a canker at waist height, and you apply the mud pack or the clay pack to that canker, there's nothing to prevent a canker from developing at 15 feet or 20 feet or 25 feet.
Right. Anthony says, I've used clay, and I think in my experience with mud packs, I mean, I haven't seen any difference in terms of efficacy of what soil you use from around the base of the tree. I've found that I haven't seen that one soil works better or worse than another from a mud pack. Do you, Mark or Laurel, know that one mud type or soil type works better than another or worse than another?
I haven't done that many mud packs to be able to answer that question.
Maybe
Eric. Different soil types.
Yeah, maybe
Eric. I really
did
think a lot of it has to do with reduction of oxygen. I mean, it's just simple as that.
Right.
Yes, the secondary metabolites do play a role, but then again, we're talking about something that's in the vascular cambium. And if you're producing a secondary metabolite that's in the soil that's adhered to the surface of the bark, well, you may affect the fungus that's on the bark surface. You're not affecting what's in the vascular cambium.
Jim English asks, how about using a spray program of competing fungal spores mixed prior to infection by the blight to establish a large population of competing fungi as a prophylaxis to ward off infection before it happens.
Never done that.
I mean, there's a long list of things we can try once we have targets. But one thing that Mark, has mentioned, we just have to make sure that whatever we're doing has the ability to get into the tree, which is where Cryphonectria is going to be living.
So along these lines, have you tried colloidal silver solution by pouring it around the root base and then having it be absorbed into the tree?
I've never tried that.
Nope.
I think there's a lot of things that people can try and see how. You just need to let us know. Try the stuff and then let us know how
See if it works.
Let's see. What about injecting fungal competitors under the bark?
It's on the list of things we can try.
It's on the list. Okay. Let's see.
Yeah.
Oxygen reduction therapies.
What would those be?
I don't know. How would
I
you
don't know
reduce? Yeah, I don't know. Let's see. There was a good one here. Here it is. Yeah. Brendan, what is the driving force between development of new compatibility types? Do some compatibility types exhibit positive survivability of the fungus compared to others? And what is the risk for introducing new compatibility types into Europe?
Well, I don't know that you're going to introduce anything new into Europe. The further east you go in Europe, the more virulent forms of the fungus you have. Like when you get to Azerbaijan, that's a pretty hot area in terms of chestnut blight. But in terms of, when I showed that cartoon that had the 8 two-celled ascospores inside the ascus, when you have sexual recombination, let's just very simply call compatibility types 1, 2, 3, 4, and 5. Of those 8 spores, 2 of them will be compatibility type 1, 2 may be compatibility type 2, there'd be 2 3s and 2 4s. So you have sexual recombination that's evident in each ascus. So you have different ascospores with different genetic makeups. So when those exit the perithecium and get blown out by the wind, you have a lot of different compatibility types being blown out of each perithecium. And that's in one ascus. And so you can have genetic recombination among all those different asci when you have genetic recombination between 2 strains of the fungus. And so what we have in the Eastern United States is just a lot of genetic recombination, and which yields genetic diversity in the fungus. In Europe, then, when you have areas that have predominantly one form of the fungus, you don't get as much recombination because the other fungi that can cross-pollinate, let's say, just aren't there in numbers. So you don't have that driving force of recombination in Europe, in some areas of Europe, than you do in the Eastern US.
All right, I'm gonna address one more question, and I think we'll wrap it up. Roland asked, I get this question a lot too. I want to get your take on it. I planted a bunch of chestnuts in 2008. Last year, a few of them started to get blight. Should I cut them down?
It's probably not going to make that much difference. I think realistically, if you cut them down, you're going to reduce the spore potential. However, the spores are still there in that orchard, and it makes you feel better by removing them, but I'm not sure it's going to have that much impact on the rest of the orchard. But yeah, I'd cut them down and remove them.
Well, and that's generally my thought. Eventually, though, you get to a point where you've just got so much blight you can't keep up with it, and
yes,
you just kind of let it do its thing.
Yes, you do.
So I mean, you can arrest it, kind of like the mud pack, or even hypovirulence. You can get it to work for some amount of time, and you can get it to prolong the life of a given tree or a set of trees, but eventually it gets overcome. At least the current stuff that we have to battle it from that side of things. I think that's why we're focusing a lot on giving the tree resistance. I mean, I think that hypovirulence, any of these biocontrols, I think these are great additional tools, but I think inevitably you need to match that with having the tree have its own set of tools to be able to fight off the fungus too.
Right. And I'm telling everybody as we install our germplasm conservation orchards in West Virginia, that if we plant 100 trees, we can expect 30 of them. They're all going to become infected, but we can expect 30 of them to sprout to the point where they flower. Is that right?
At least, I mean,
30%?
it's my experience that we get about, so I've seen orchards that are 30, 40, almost 50 years old of wild type Americans. And we get on a good site with good maintenance, you don't let the deer in and you don't
Right.
let weeds run wild. But we get about 50% survival of the sprout clumps long term. We get flowering of virtually all of those trees at some point. Even the second flush of sprouts, third, fourth, fifth flush of sprouts
Right.
can and will flower. So you can have these GCOs or wild type Americans survive for a long amount of time. I think hypovirulence will help that, or mud packing. You can extend the longevity again of individual trees or that site with those tools, but I don't think that they're absolutely necessary for prolonging their lifetime or
Okay,
getting them to flower. All right, thank you, Mark, Laurel. Thanks for hanging in there. As always, we got amazing questions. I really appreciate you guys for hanging in there too, and asking some really thoughtful and providing some cool things that you could try and let us know how it worked out. Before I turn it over to Lisa, I'm going to put a plug in for next month on May 21st, I think is our next date. I think you're going to hear a lot from me. My plan is to talk a lot about site selection and planting. We get a lot of questions about how do you plant chestnuts? How do you select the best site? So my current plan is for me to do some of that and have another guest. So if you'd like to hear me talk, tune in next month. You'll get a lot of that. And with that, I'm going to turn it over to Lisa. Thanks again, Mark and Laurel. It was a real pleasure.
Yeah, I don't have much to say in conclusion, except to add to Sarah's thank you to Mark and Laurel. I know that it takes a lot of time to prepare presentations like this. I also want to thank all the participants. When Sarah and I dreamed this up a year ago, we had no idea it would have the staying power and the interest that it has, but we hope to continue Chestnut Chats indefinitely. There's just no shortness of ideas and topics when it comes to chestnut. So I think we're all going to be continuing this, and hope to see you all in person soon, and stay safe and get your vaccines. That's my only shameless plug. Take care, everybody. Have a great weekend.
Okay,
Thank you so
bye-bye.
much, Mark and Laurel.
Bye-bye.
Thanks, Sarah.
See ya. See you in May, guys.